Hey guys, today this video will be all about pedigree and environmental influence. Let's start with pedigree. I've drawn here a pedigree for dogs. You have a legend here, so the males are squares, the females are circles. Pretty accurate. So that's a female, that's a problem. That's what it is. Let me fix that. This is a male, right? And then that's a male, that's a female, that's a male, that's a male, right? Then the coloration tells you if a puppy, those are dogs, have anery, exhibit anery or not. Anery is anery, A for the lack of, and tail, so it's a puppy with no tail. So if they don't, if they have a tail, if they don't exhibit anery, they're in blue. And if they don't have a tail, exhibit anery, they are in red. The pedigree of that family shows that a female that is not affected with a male that is not affected, individual 1 and 2 got offsprings, puppies, one female that is not affected, and a male that is affected. When we look at that pedigree, the first thing we can say is, oh, those two individuals must be heterozygous, right? If we're talking about anery, and I'm going to draw now the genotype of those individuals, it will use the letter A, it has to mean that those individuals are heterozygous. This is the only way for the male here to get the anery, right? It's the only way for that individual, because anery is a recessive disease, it has to be like that. Well, how do I know it's a recessive disease? Well, the point is when you look at this, and you see that not all offsprings have it, and the parents don't have it, it has to be recessive. If it was dominant, if an offspring exhibited, a parent is exhibiting it, because the parent is carrying the dominant gene. So if the parents don't exhibit it and an offspring exhibit it, it means it's recessive disease. And it also means it's heterozygous parents. Can it be both parents being heterozygous? Well, let's look at it. If you have two capital A and one A, big A, little a, you cannot have, if that was capitalized, you cannot have a puppy with two little a's. You cannot. The dad can give in 50% of the case the little a, but the mom has to give the big A. That's why you know it's heterozygous. So you see how I use that pedigree to actually characterize the disease type, recessive, the parent's genotype, and the offspring genotype, like that. Now, you don't know if that individual here, that female, got two big A's or one big A and a little a. It can be either one, right? Because she doesn't have the disease. So that's the two types, right? That individual here is coming from outside of the puppy family, has the disease, so we know that male has also little a. And now the mystery I'm asking you is, what is the probability of that puppy to have that disease? That one here, the male puppy from that couple. You have to work through that, right? So if you want to pause the video here and just take a stab at it, and then I'm going to tell you now. Nice opportunity to pause, okay? So the mystery is solved this way. We know that the puppy here is definitely inheriting a recessive allele from the father, right? So the puppy has definitely big little a here. The question, it's all about that allele from that mother, right? So if the mother has one of recessive allele, that means that that puppy has 50% chance if the mother is heterozygous, meaning that if it's this here, let me use black here. If that's the case here, there's 50% chance that that allele will be little a. So it means that in 1.5 chances, if the mother is heterozygous, the puppy will have anery here. Now, if the mother is not, she won't pass a little a, so there's 0% chance of having anery. Right? So it all depends on her genotype. So that's why, and these are dogs, right? But if we talk about human beings and any other disease, the pedigree can be the same, the layers can be the same. Now you see the power of for that female individual to be, I need to get genetically tested to know if I'm heterozygous or homozygous because my partner has the disease. And if I'm heterozygous, there is 50% chance a child is going to have the disease. Depending on the disease, that's a big decision. So genetic typing, genetic tests are really handy for couples when one knows has a disease and the other one doesn't know if it's a healthy carrier or not. Especially if that female knows that one of the brother or sisters have the disease, right? So that's the part about pedigree. So let's take a break. Just before we go fully blown on break, you need to be able to infer all those things from pedigrees, and as you can tell from the PowerPoint, pedigrees that we work with can be more complex than that. But if you work your way logically, you can do this. You can infer heterozygosity or homozygosity. You can infer the genotype from the pedigrees. Okay, let's switch to environmental. Guys, I'm so proud of myself. This is the cat. So on slide 58 of your slide stack, you have the example about the Siamese cat. And the Siamese cat, on the parts of its body where it's colder, which means simply the tips, right? So the ears, the muzzle, the nose, it has, and the face, it has some dark coloration, simply because the genes in low temperature actually don't, uh, sorry, they are active only when it's cool. When it's too hot, because the cat is an endothermic animal, which means it produces its own heat, which means that all the other part of the body, closer to the organs, produce heat. So that heat inactivates the genes, but in the colder part, the genes is activated. The genes are activated. So now we know that influence. Theoretically, if you take a Siamese cat and you stick to it a cold ice pack, and you wait enough time for enough mitosis to go through and having the entire patch of skin here going through cell divisions and having gene expressions going on, then theoretically, if you keep that ice cold and you remove it after whatever time it takes, maybe like, I don't know, a week to have the renewing of the cells in human beings in 30 days, all their cells have come anew and multiplied. So in cat, maybe a week, maybe 2 weeks. Turns out that patch of skin below it will be dark, will be black, because the genes will activate now that it's cool enough. And that's what it is, environmental influence. That's one type of environmental influence. Don't do that to your cat, please. That's kind of cruel. But the theoretical exercise is interesting, right? Other environmental influences come from your soda can, the pulp. On it, you can see phenolphthalein warning. It's because some of us have rare conditions that if we drink phenolphthalein or we get exposed to phenolphthalein, it's poisonous to us. I'm not one of those, but some of us in humans beings have that. And that's another environmental influence. Not exposed to phenolphthalein from any sources, fine. Drink it because it's in soda, die, right? And that's the environmental influence. Some genes will be turned on or off, or some, and it's really talking about genes, right? Phenolphthalein is not poisonous. We can drink it all day, but people without the condition, it's not poisonous. It's only when you don't have the means to deal with it because you don't have the right genes, right? So environment can alter the phenotype. Environment times genotype equal phenotype. That's why you see that's G by E equal P. Genotype times environment equal phenotype. That's why you can always know your phenotype, because that's what we see, but you don't always know the genotype behind it. Because depending on the environment, you can have the same phenotype, but two people with the same phenotype can end up there with different ways. There are different genotypes and different environments, right? For example. So here you go. That's the environmental influences. See you next time.